And then so they let this, and they stimulated it to allow it to divide, so this would mimic the act of fertilization. And so an embryo starts to divide. And then similar to what is done with making transgenic mice, these cells were then implanted into the inner cell mass of a blastocyst, and then this whole blastocyst was transferred to a surrogate mother. And after the normal gestation period, you got the birth of this lamb that was basically created by nuclear transfer. So this shows that the nucleus of the udder cell can be reprogrammed into express embryonic genes once it's fused with the oocyte, and that this can actually result in the production of a fully formed embryo. Okay, so that's somatic nuclear transfer. Another set of experiments that show that gene expression is controlled by something in the cytoplasm are cell fusion experiments. So it's pretty easy to fuse cells together. You can do this in the lab using chemicals like polyethylene glycol, or you can use viruses. Some viruses tend to cause cells to fuse together. And in the end, you have two cells that fuse together. They have their separate nuclei, and now they're sharing a common cytoplasm when they're fused. So that's one of the experiments that was done is to fuse a human liver cell with a rat muscle cell. So you can, in these experiments, you're able to tell which proteins are from the human and which proteins are from the rat. So if you fuse the liver cell with the rat muscle cell like they do here, now the cytoplasm between these two cells is shared. And actually it's shared throughout the myotube because remember these cells are all fused together. And so what happens is when this liver cell from the human is fused to the rat muscle cell, what was observed was that actually human muscle genes were expressed in this new fused cell. So this shows that something in the cytoplasm of the muscle cell is activating muscle-specific genes, even in the human nucleus that didn't come from a muscle. So it's basically reprogramming what's being expressed from that human nucleus. So this shows that there are cytoplasmic factors that can control gene expression. Similar to what was shown with the somatic cell nuclear transfer. And this also shows that, again, gene expression is plastic. Once you have the expression of liver cell genes, doesn't mean that they can't turn on anything else. Okay. So another piece of evidence that shows that gene expression is plastic is the phenomenon called transdifferentiation. And this is the case where a cell of one differentiated cell type can change into another one. And this usually occurs in cells that are derived from developmentally related cell types. So for example, one type of cell that's derived from the ectoderm can turn into another type of cell that's also derived from the ectoderm. So for example, there are cells in the eye. This is showing a pigmented cell that's derived from ectoderm. And you can grow these pigment cells in culture and get a layer of them on your dish. And if you change the culture conditions, maybe by just changing the growth factors, changing the signaling that is occurring in those culture dishes, you can get changes or transdifferentiation of those pigment cells to more retinal and lens type cells. And normally the retinal cells and the lens cells are also derived from ectoderm. And if you keep on changing the conditions, you can even make them change into lens cells. So you can have basically 3 phases of transdifferentiation. Again, starting with a somatic cell and just changing the signals that it's receiving. Okay. So this is important to say if it's hard to make new lens cells, if you need more lens cells, maybe you can treat your eye with something that would just change those pigment cells into more lens cells. Okay, transdifferentiation has also been observed in other cell types, not just the eye. So there are cells derived from the neural crest called adrenal chromatin cells, and these express adrenaline, and that's what's shown here in these vesicles, okay, in these vesicles. Okay, and then if you culture these in vitro and remove glucocorticoids from the culture media and add nerve growth factor, you can change the phenotype of these chromatin cells into more sympathetic neuron type cells. And this is interesting because they express a different kind of adrenaline. They express noradrenaline, and both of these cells are normally derived from the neural crest. So you can induce transdifferentiation into different cell types this way. And then it's not only observed just in ectodermal cells, you also can see it with cells from the other germ layers like ectoderm. So here, the pancreas and the liver are both derived from endoderm. And if you take a transcription factor called PDX1, which is a pancreatic specific transcription factor, so it's expressed mainly in pancreatic cells, and you express this transgene in a liver cell, you can convert that liver cell into a pancreatic cell. And this is shown in these experiments where this is zebrafish. So this is developing zebrafish. Here in L, in red, is where the liver normally will develop, and P signifies the pancreas. And in this case, the pancreas is stained with GFP. On the bottom, they're showing you a zebrafish that has had liver cells that have been induced to express PDX1. And what you see after several days is that this liver, where the liver is supposed to be, you start to see ectopic pancreas. That's what EP is. So in addition to the pancreas that it naturally has, the liver cells have been converted into pancreas cells, and we know this because they're expressing GFP under the control of the PDX promoter. So here, just expressing a cell-specific transcription factor can convert one cell type to another cell type. And this type of strategy has been proposed as a way to make more pancreatic cells, say for people that have autoimmune diabetes or need to replace pancreas type cells, which are pretty hard to do, but it's easy to get lots of liver cells. So this is an example of transdifferentiation. Okay, so a lot of the developmental biology concepts that we have discussed can be applied to the medical field and in the clinic. And in particular, in recent years, you may have heard a lot about the use of embryonic stem cells for regenerative medicine